Hello, good afternoon. Today in my video, I'm going to talk about biofilms. And just an introduction, my name is Sanjay Suresh. I'm a student taking biomedical science microbiology at the University of New England. As given by the instructions, here is the set of notes that I've been using for my presentation. Today in my presentation, I'm going to be talking about biofilms and what are some ways to eradicate them, what are they, what are some key characteristics, etc. To start my video, what is a biofilm? So a biofilm is a thin, slimy layer of microorganisms. These are mainly bacteria which stick and adhere to a surface to each other, and they are embedded in a self-produced matrix. So basically, these are like community of bacteria that live and stick together. So, what are some key characteristics of biofilms? So, biofilms, mainly some key characteristics is attachment. So, some of these microbes, they adhere and they attach to the surface of teeth, rocks, pipes, and even medical devices. One other main characteristic is protection. So, what are some protective mechanisms? So, they produce a sticky matrix that shields them from dying out. Also, communication. So, just like how humans talk to each other, these microbes can also talk to each other chemically, but in a chemical way. They coordinate with each other. And another important characteristic is resistance. So biofilms are way harder to kill compared to free-floating bacteria. So these are some of the main key characteristics. So one important thing that these biofilms use is that they use a protective matrix which blocks penetration of antibiotics. So how this looks is it is mainly like a slimy layer that acts like a physical barrier, and it leads to a slow diffusion of antibiotics. It mainly causes them to trap and neutralize, and it shields the bacteria from immune cells. Also, these are mainly slow growing, and they're dormant bacteria. So inside the biofilm, they have the nutrients. The nutrients and oxygen are very limited. So many of these bacteria, they become slowly growing, and they're mainly dormant. So the antibiotics or drugs, when they target them, they become less effective. And so this is mainly some of the key characteristics of the bacteria, and also they also alter gene expression too. So now in the next part of my video, I'm going to talk about polymicrobial biofilms. So what are these? These are biofilms which are made up of multiple different species of microorganisms that live together in a community. So just like how humans live together in a neighborhood, these bacteria also, they live in a community, and they contain multiple different type of organisms. For example, you have bacteria, you have fungi, viruses, and archaea. And what are some examples of these polymicrobial biofilms? So one is dental plaques. So typically, when we have plaques in our cavity, they contain many bacterial species which are associated with the cavity and gum diseases. And also another example of a polymicrobial is chronic wounds. For example, in diabetes, foot ulcers, which contain many bacteria that live together. So the next part of my video, I'm going to talk about single species planktonic models and why they're insufficient. So why are they insufficient? Mainly because of real infections, which are mainly polymicrobial. So many of these infections, they involve the interaction of multiple organisms rather than one species. For example, in dental plaques, chronic wounds, or lung infections like cystic fibrosis and catheter-induced infections. And inside the body, they mainly encounter many of the immune cells, mucus, neutral gradients, low oxygen species, and mechanical forces. So yes. And now in the next part of my video, I'm going to talk about tactics which are currently being developed to eradicate these biofilms. So one important strategy is the antimicrobial peptide. So these are proteins that are made up by many of the living organisms. They're made up by many of the living organisms which are a part of the immune system. For example, you have bacteria, fungi, and viruses. Okay, so another part that we have here is we have the dispersal methodologies. So these are strategies which are used to break up and remove and trigger the release of these microbes from the biofilm community. So one example is the physical dispense methods. So what these do is that they disrupt the biofilms. For example, if you scrape or brush, they can disrupt the biofilm communities, or ultrasounds, or fluid shearing or flushing, debridement of wounds, or surface vibrations. And we also have chemical dispersing methods, which are detergents. For example, we have or certain antiseptics. For example, we have chelating agents. So what chelating agents do is that they remove metal ions that stabilize the biofilm. For example, we have EDTA. We also have oxidizing agents, which also damage the microbial cells and matrices. For example, we have hydrogen peroxide and we have bleach. Also, we have enzymatic dispersal methods, which can digest certain components of the extracellular matrices. For example, we have the DNase, which breaks the extracellular DNA. We have proteases, which help digest proteins, and then we also have glycosides, which help digest the polysaccharides. And another important aspect that I want to talk about in my video is the EPS interdictions. So EPS basically stands for extracellular polymeric substance. And this is basically like a sticky protective matrix that surrounds the microbes in the biofilm. It acts like a scaffold and shields the microbial community. So what we do is in this interdiction these are basically strategies that disrupt and prevent and weaken and degrade the substance. For example, we can use DNase 1. Many biofilms use eDNA as a structural mesh, and this DNase, it cuts this DNA apart and it weakens it. For example, you can use them in lung infections, cystic fibrosis, chronic wound infections, dental plaques. Another method that we could use is this dispersin B, which targets certain type of biofilms. And what it does is it hydrolyzes the matrix polysaccharides which are involved in the bacterial adhesions. Like that, we have other enzymes such as alginate lyase, which is a major EPS polysaccharide. So it disrupts this, and it's mainly formed by Pseudomonas aeruginosa. So in the next part of my video, I'd like to talk about nanotechnology. And what is nanotechnology? It's a science and engineering which works. It's mainly the science and engineering of the work and with material. We use a nanometer scale, which typically ranges from 1 to 100 nanometers. And this can behave very differently than a larger scale. So, example of nanotechnology I can give is nanomedicine, which basically it's used in medication, so it can help to deliver the drugs, and it can help in imaging, it can help in cancer therapy, it can help in anti-biofilm treatment, and these can carry antibiotics deep into the biofilm structures. So the last part of the eradication I'd like to talk about is phage therapy. And basically phages they use bacteria. They use a bacteriophage. And what a bacteriophage is, it's a type of virus that specifically infects the bacteria. So unlike the antibiotics, which are chemicals, the phage therapy, it uses biological agents that can replicate at the infection site, as long as the susceptible bacteria are present. For example, we can use multi-resistant acinetobacterial infections. So the last part, and also I'd like to talk about quorum therapy. So quorum therapy is, it mainly disrupts and blocks the communication system of the microbes. And what it does is interferes with the signaling. So basically it helps to reduce the virulence, it impairs the biofilm formations, it increases antibiotic susceptibility, and it reduces toxins. And so to wrap up my video, I'd like to talk about what critical areas of biofilms. I'd like to talk about one of the critical research areas of the study in quorum sensing and quenching mechanisms. So basically, what are biofilms? So biofilms are highly resistant to antibiotics and immunological responses because the bacteria live within them for communication and coordination. So if we want to understand how these signals regulate, we need to talk about the virulence factor production, antibiotic resistance, and dispersals. So when we talk about research about quenching, it's especially important to know it because it offers an alternative to traditional antibiotics. So instead of killing the bacteria directly, quorum quenching approaches to disrupt the bacterial communication. So for example, it prevents the biofilm maturation, it reduces their pathogenicity, and it increases antibiotic susceptibility. And they're at low risk for resistance development. For example, you can, promising research directions, they include enzymes that degrade the signaling molecules, synthetic quorum sensing inhibitors, plant-derived anti-biofilm compounds, and bacteriophage-based disrupt systems. So what are some of the challenges that this area in this particular area? So example are chronic wound infections, cystic fibrosis, implanted medical devices, dental plaques, and industrial biofuelings. So I really hope you like my video about biofilms, and please do subscribe to my channel. Thank you.